So today we're going to be talking about transformation. Okay, so transformation is the introduction of DNA into the bacteria without the use of a carrier. And this process begins with the following. You mix the bacteria and the DNA together and you incubate this on ice for 30 minutes. All right? Then after 30 minutes, the tube is taken and incubated at 42 degrees, and this is called a heat shock for exactly 90 seconds. No more than 90 seconds, or you will fry the bacteria. And then this tube is returned back to ice, and it is incubated on ice for about 2 minutes to cool the sample down. And then to this tube, 800 microliters of L broth is added. Okay, and this is incubated with shaking at 37 degrees centigrade for 30 minutes and slow shaking of the sample. All right. After this step, then you plate down 2 aliquots, 10 microliters on one plate, and then you plate out, let's say, 50 microliters on another plate. And this is, of course, where your L-amp plates that you made up a few days ago, and they're stored in a refrigerator. And these plates are incubated at 37 degrees centigrade overnight. Okay? And this is the process of transformation. So let's go back and let's just talk about this. The first step is mixing the bacteria and the DNA in a tube and incubating on ice for 30 minutes. The purpose of this step is simply to bring the reactants, so to speak, together, very close together, because the next thing that happens is you heat shock the bacteria. Now, the bacteria that is used is competent, meaning that its cell wall is destabilized. And so when you heat shock the bacteria in the presence of the DNA at 42 degrees, it facilitates the transfer of the DNA pushed into the bacteria. And so the next step is, after you push the DNA into the bacteria, then you cool the sample back down on ice for 2 minutes. And that is, you allow the cell wall to close the pores that might open up during the heat shock. After the recovery, and then you add 800 microliters of L broth to the sample. And the purpose of this step is that you have transformed the bacteria, you have heat shocked it, and then now the bacteria is not very healthy. And so when you add 800 microliters of very rich media, sometimes we use SOC broth, very rich, and you shake slightly at 37 degrees for 30 minutes, you allow the bacteria to recover. And the reason why you need that recovery is that the next step, which is you're going to plate aliquots, a small aliquot, 10 microliters, and a 50 microliter aliquot on plates, agar plates that contain the appropriate antibiotic, ampicillin, at 100 micrograms per mil, right here. Okay? Now, when you plate the bacteria here on this, only the bacteria that contains the plasmid will survive. And the reason for that is that plasmid normally carries with it a antibiotic resistance gene in it. And so the expression of this protein then will allow any bacteria that harbors this plasmid to survive on agar plates. So when you plate this out, and whichever colonies are survived means that they contain the DNA of interest. And so this is the process is incubated at 37 degrees overnight, and when you come in the next day, you're going to see a plate, hopefully, that will look like that. It's just a few of the colonies, okay, like so. And so if a colony survive right here on the AMP plate overnight, and so you know that each one of these colony has the plasmid in it, because the plasmid carries the resistant gene which is expressed in this bacteria, thus allowing them to grow on agar containing ampicillin. And so this is going to be our lab on bacterial transformation.